Okay, good morning, everyone. My name's Matthew Self. I'm a researcher from the Netherlands Institute for Neuroscience in Amsterdam. And today we're going to be talking about benchmarking a laminar fMRI. And the delicious cake on my title slide gives you a hint as what we're going to be talking about. We're going to be talking about layers, but not layers of a cake, but rather layers of cortex. In particular, we're going to be interested in one of the key questions in advanced fMRI, particularly in high field strength fMRI, which is whether we can use fMRI as a tool to investigate the processing which is going on in the different layers of cortex. So laminar fMRI is a relatively recent tool, which has been made possible as voxel sizes have been pushed into the sub-millimeter range. So with a voxel size of 0.8 millimeters isotropic, that's about the same size as layer 4, for instance, in the primary visual cortex. So this opens up the possibility of actually isolating signals, fMRI signals, in different layers of cortex. However, there are a number of complications to analyzing laminar signals, and particularly laminar bold, which has been the main technique that's been used. So here's just a list of some of the difficulties in interpreting laminar bold signals. So for example, although sub-millimeter voxel size is a technical feat, it's still not quite small enough to be able to isolate signals from a single layer. Okay, so the layers are generally smaller than 0.8 millimeters, and so your voxel is always going to contain signals from multiple layers. Also, if you look at something like, in particular, gradient echo bold, then this signal is really biased towards the large surface veins on the surface of cortex. And so that's going to distort the laminar profile that you'll measure, and your bold signal is going to be increasing as you move towards these surface veins. The blood flow in cortex tends to come into the deep layers and move up towards the superficial layers. That could also be an issue in bold signal generation, because the bold signal that's generated in the deep layers is going to bleed through into the superficial layers. Let me smear out any laminar profiles that you're trying to measure. And there may also be biases in the bold signal towards layers which have particularly dense microvasculature. So for instance, we know that layer 4 has a very dense capillary bed, and we'd expect the bold signal therefore to be larger in that layer. It's also a very tricky technique to analyze. So cortex is not just a uniform flat sheet. It's convoluted into sulci and gyri, and the layers have different thicknesses on the crown of the sulcus and the depth of gyrus. So that means that you need to use sophisticated analysis techniques or computational models to work out how you isolate signals from different layers. Finally, the link between neural activity and bold signal is really only partially understood, which means we don't know exactly which neural processes in different layers are going to be contributing to our bold signal. So I think given all those complications in analyzing laminar data, it's really critical to be able to cross-reference or cross-validate the bold signal that we measure with laminar fMRI to the known laminar differences in neural activity that we can observe in animal studies. So that's what the focus of this talk is going to be. We're going to be looking at benchmarking laminar fMRI by looking at EFIS studies in macaque monkeys primarily, and then comparing it to recent fMRI studies that have been carried out around the world. Okay, so I'll start with a little intro into the laminar organization. And throughout my talk, I'm going to focus on the primary visual cortex. And that's for a number of reasons. The primary visual cortex, V1, it's the most heavily studied area, both in the EFIS studies and also in the laminar fMRI studies. It also has perhaps the most intricate laminar structure. So you have this very specialized layer 4C, which you see on the slide here, with this very cell-dense layer. So it provides sort of ideal testing ground for validating the MRI studies. If we can pick up the layer differences in primary visual cortex, then we should be able to pick them up in other cortical areas. So what we see here is just an anatomical section of V1 of the macaque monkey. It's divided into different layers. In particular, has this, as I said, a very dense layer 4C. You have deep layers 5 and 6, superficial layers layer 2, 3, and the cell sparse layer 1 at the top. And the thickness overall is between 1.5 to 2 millimeters in the monkeys. In humans, it's larger than that, it's thicker than that. But what we see is that we know quite a lot about the inputs and outputs of this area of cortex across the different layers. So if we focus first on the input side, we know that the feed-forward connections coming up from the retina through the thalamus are terminating very heavily in layer 4C, also to a certain extent in layer 6 and layer 4A. whereas the feedback projections coming back from higher cortical visual areas, they tend to avoid layer 4 and they terminate largely in layer 5, in upper layers, layer 2, and in layer 1. So this is clear anatomical segregation between the feed-forward projections and the feedback projections. And I'll come back to that on my next slide. Also on the output side, we see a clear segregation as well. The upper layers, layer 2, 3, and layer 4A, sorry, layer 4B, they project to higher cortical areas, whereas the deep layers, like 5 and 6, they project to subcortical areas such as the superior colliculus or back to the lateral geniculus. Okay, so both on the input and output side, we have a separation of the different layers, and we think the different layers are really carrying out different functional processes during visual processing. Now, just to focus in again on the difference between feed-forward processing and feedback processing across the different layers, is maybe the most dramatic difference between the different layers. So here we have two slides which were originally published in the 70s. On the left-hand side, we see the feed-forward projection. So this is autoradiography, and they're looking at the projections coming from the LGN terminating in V1. And what you see is this really, really strong projection to layer 4C. Okay, so it's very clear and dense projection, also a little bit to layer 6 here and a little bit to 4A. This is white matter. If you look, on the other hand, at the feedback projections coming from higher cortical visual areas, this was a study from Kathy Rockland where she injected a tracer in V2 and then looked back at where the V2 axons are terminating in V1. Then what you see is this very, very dense projection to layer 1, and this white stain here, and also a little bit to layer 5. This is also white matter at the bottom. Okay, so we see this really, really clear segregation in the anatomy. There's also a really, really clear segregation in the function of these connections. They perform quite different roles when it comes to cortical processing. So we know that feed-forward projections, they drive neural activity, and they're largely responsible for the size of the receptive fields, and they're also responsible for the tuning properties of the receptive field. For example, we know the orientation tuning of a cell in V1 comes from the arrangement of its feed-forward projections that it receives. There's also inhibition in the feed-forward pathway, so things like cross-orientation inhibition, also through targeting of local interneurons or through presynaptic processes. Feedback, on the other hand, fulfills quite a different function. So feedback, it can drive neural activity, but mostly it's modulating ongoing neural activity in the cortical circuit. It plays an important role in integrating global contextual information from the entire visual scene with the local information that is present in the receptive field. So we have phenomena such as figure ground modulation or surround suppression, and I'll be talking a little bit more about those in a couple of slides. Feedback allows you to select behaviorally relevant items and modulate the processing of those items. So we're talking about attentional selection, and also to maintain items in memory when the item is no longer present in the visual scene. And I'll come back to that at the end of my talk. Finally, feedback is thought to carry predictions about likely receptive field content, and it's also responsible for a lot of the rapid learning effects that can take place in the primary visual cortex. So there's a clear sort of division between setting up the receptive field and modulating the receptive field to produce more sort of cognitive processing through the feedback processes. So what I'm going to do, I'm going to focus first of all on the feed-forward driven activity. I'm going to look at how feed-forward activity varies across the layers of V1, and then we'll go on and have a look at feedback processes. So starting with visually driven activity, I'll take you through a study we published a few years ago now, in which we had monkeys, awake monkeys, and we had a laminar electrode implanted in V1. So this is an electrode with multiple contacts arranged vertically, allowing us to record neural activity from every layer of cortex simultaneously. I'm going to start by focusing on the high frequency activity, the spiking activity that we can pick up from each of these contacts. So here you see a laminar plot. This is just the response to a high contrast checkerboard, just a very strong visual stimulus, which should drive activity across the cortical column. And the measure we're looking at here is multi-unit activity. So we use a measure in the lab which reflects the average spiking activity within about 100 microns of the electrode contact. And that's quite a nice measure to compare to fMRI. It's not biased towards particularly high amplitude spikes. It's just giving you the average spiking activity around here. So that's what you see in the color code here. So this is depth. 0 is the boundary between layer 4 and layer 5. And then we have the deep layers and the superficial layers at the top. Time is on the bottom. And if we average across the layers at the top here, so if we just average this way, then you see the visual response starts with a very strong peak, which then tails off towards spontaneous activity over about 300 milliseconds. If we average across time, so if we look across the layers, then what we see is we get the strongest multi-unit activity in response to a checkerboard around layer 4C, so around the boundary of layer 4C and layer 5. You can see as you move towards the superficial layers, this response tails off and becomes much weaker in the superficial layers. Now this is interesting. So this is a multi-unit signal, right? We're measuring signals which are just an average of the spiking around the electrode contact. So the fact that the signal is weaker in the superficial layers, it could be that just spike rates are generally lower in the superficial layers. It could also be that there's just a sparser signal in the and that you have fewer cells responding, which may result in just a lower level of the signal overall. Okay, so, but this is the general laminar profile. You see this very large peak response centered on layer 4C, tailing off. What's kind of interesting, you can't really see in the color plot here, but there are quite large differences during this sustained period of activity here. I'll just isolate those on the next slide. So if you look here, so here we're letting the peak go by, and now we're just looking at this sustained period of activity here. So the time actually starts at 150 milliseconds on this graph. And what you can see is this very obvious blue stripe in the graph. So this is layer 4C. And if you look at the color scale, what's happening here is that layer 4C responds very, very transiently to the visual stimulus and then goes back to baseline for the duration of the stimulus presentation. So there's a very transient response in layer 4C, whereas in the superficial layers and the deep layers, there is sustained processing of the visual stimulus. You can see spiking carries on for the duration of the visual stimulus. Now, this has relevance for laminar fMRI studies is because the laminar profile you're going to get is going to depend upon the visual stimulation pattern you use. If you use very strong, transient, heavy stimuli, where the stimulus is refreshing a lot, that's going to drive this peak activity in layer 4. And so you're going to get a bold profile which is dominated by layer 4. However, if you have a very sustained visual stimulus, which doesn't change very much, then that's going to bias you more towards the superficial and deep layers. Okay, now we know we've been looking at spiking activity, high frequency activity, but we know from previous studies that the bold signal actually correlates more with low frequency activity, so more with the local field potential. So it's interesting also to look at the local field potential across the different layers. So actually the local field potential itself is not super interesting because it doesn't vary much across the layers. It contains global components, which are really the same in all the different layers. So what we tend to do is transform the local field potential to reveal local information. So we do that as we generate current source density, which is the second spatial derivative of the LFP. Right, so that gets rid of the global component and it isolates the local components. Okay, so if we look at the low frequency activity, the current source density, then we get quite a different picture. So again, it's time and depth, and again, it's the response to a checkerboard. But what you're seeing now, so these red colors are current sinks, so that's current flowing into neurons. That's generally excitatory current. Whereas these blue colors here are current sources, and they're largely current being passively returned to the extracellular medium, could also be active inhibition of a particular layer. And if we look at the amplitude of these different sinks and sources that are produced when a checkerboard is shown, first of all, you see this little sink here in layer 4C. So this is the input arriving from thalamus, terminating in layer 4C, opening up ion channels in layer 4C and producing spiking activity in that layer. But you can see the amplitude of the current sink is actually not that great in the CSD. The CSD is actually dominated by these long-lasting, sustained current sink source pairs in the superficial layers and by this long sink in layer 5. So what we see then is quite a different profile, with the MUA being dominated by layer 4 and the CSD being dominated by the superficial and deep layers. If we put those two next to each other, here's the multi-unit activity, here's the current source density. We get quite a different view of the laminar profile. So how does that compare to the profiles that we see with laminar bold signals? So there are two studies. This figure actually comes from the Chen et al. Study, but there was a very similar profile published earlier in Koopmans et al. Study. And in both of these studies, they looked at the responses to flickering checkerboards. And what they found, first of all, is that the response actually increased towards the superficial layers. These are the superficial layers. These are the deep layers. And they use techniques which actually remove the contributions of these large draining veins on the superficial surface. So if you do that, if you get rid of the contributions of those large veins, then that reveals a laminar profile which peaks in the middle in layer 4. So here you see then a laminar profile from bold fMRI, which is much more similar to the spiking activity that we see. So this initial large response in layer 4, than it is to the synaptic activity measured by current source density. So that's quite interesting. So the visually driven laminar bold profile seems to resemble the spiking activity that we see in cortex. And there may be some reasons for that. It might be related to the stimulus choice. As I said, if you use flickering checkerboards, that might drive a more layer 4 dominated profile. On the other hand, it might also reflect a bias of the bold signal towards layer 4. So we talked about layer 4 having a denser capillary bed than other layers. So the bold signal might simply be higher in layer 4. Or it may even reflect a difference in coupling between neural activity and bold across the different layers of cortex. That still remains an open question to be addressed in the future. So one way to get away from possible biases of the bold signal to particular layers of cortex is to not look at the absolute level of the bold signal, but to look at things like tuning properties. So for example, you can look at receptive field size, and you can measure orientation tuning. And these things you can measure regardless of the overall activity level. As long as the activity level isn't 0, you can measure the receptive field size. So if you do this, it doesn't depend upon the overall activity level of the signal. And you can see here from our laminar study from the monkeys, we see two things here: the receptive field size in red. So just we used a drifting bar to measure and map out the receptive field, and the orientation tuning in blue. So these are just responses to drifting ratings. And what you can see in both cases, so for the receptive field size, there is a smaller receptive field around layer 4C, and the receptive field size gets much, much larger as you move towards layer 6. And this is a finding which really echoes work going all the way back to Goebel and Wiesel, showing that the input layers, layer 4C, has the smallest receptive field size. Similarly, for the orientation tuning, we see a clear dip in the orientation tuning strength around the boundary of layer 4C and layer 5. So the cells here are more weakly orientation tuned. The cells, for instance, in the superficial layers. And again, that is also something that Goebel and Wiesel showed back in the 60s, but this shows it nicely simultaneously across all different layers. Now, what happens if we compare that to fMRI signals? Now, as far as I know, no one has carried out a laminar fMRI study looking at orientation tuning strength. I think that would be pretty challenging, maybe not impossible, but it's not been done. On the other hand, there was one study from Fracasso et al., so this from Sergio Dumoulin's group, where they looked at receptive field size or population receptive field size across the different layers of V1. And what they saw, so just to explain their x-axis here, this is distance from the gray matter, white matter boundary. So this would be the deep layers here, go through the superficial layers here. And what you see is, again, higher or larger receptive fields in deep layers, coming down to a dip around the middle layers, so layer 4, and then increasing as you move to the superficial layers. So although this is a lot noisier than what you can measure with electrophysiology, maybe because of these partial volume effects that we have in fMRI, there's a really clear correspondence between the shape of the two red lines using the two different techniques. So using neural tunings or avoiding using the absolute bold signal may be a nice way of getting rid of biases that are present in the bold signal in particular. So I think that's a promising technique. Okay, now I want to move on and look at feedback or top-down effects on visual processing. And I want to start with figure ground modulation. This is a paradigm that we've used for many, many years to isolate top-down processes. In this paradigm, we have an awake behaving monkey who is fixating on a small red dot. Then we show a visual stimulus, which is full screen and consists of lots of small oriented lines. At one point in the visual scene, we present a figure, which is made of lines of the opposite orientation, so it pops out. And we are recording from cells which have their receptive fields located represented by the blue circle here. Okay, so that's where we're recording from. So in this case, the receptive field is actually on the figure, but we can also put the figure somewhere else, right? So the monkey is still fixating, the figure is up here. Now our receptive field is on the background. And we can look at the difference between the figure and background situation. Ultimately, the monkey is going to make an eye movement towards this figure. So here we can look at the effect of context. So here the receptive field is on an interesting figure, it's behaviorally relevant. but the information that tells the receptive filters on a figure is outside the receptive filter, right? So it's a contextual effect. Now, despite that, we see a clear difference in neural activity in V1. So time 0, the stimulus turns on. This is kind of a cartoon showing the results of many, many different studies. But you see initial burst of activity, which is the same on the figure and on the background. And then at 100 milliseconds, we start to see an increased neural response on the figure, and actually a weaker neural response when the receptive field is on the background. So that leads to this modulation of neural activity, this gray region here, which we call figuregram modulation. And we've shown in other studies that this modulation comes from feedback from higher visual areas, and it strongly correlates with the percept of the animal. Now, figureground is a nice paradigm because it can be carried out in lots of different species in pretty much exactly the same way. So we've shown these figuregram modulatory signals in mouse V1, in monkey V1, in human V3 as well. So I'm a little bit obsessed with it. But this was a case where we had an electrode, we had an epileptic patient who had electrodes implanted as part of her treatment in the visual cortex. We were able to measure spiking activity in that area. And we see very, very similar figuregram profiles in all three species. So it's a nice paradigm to compare across different species. Now, what we see if we look at the figuregram modulation across the layers, so this is just averaged across all the layers, but if we actually split out the figuregram modulation into the different layers, then what we see is this laminar profile here. So this is figure minus ground. Again, depth is on the y-axis and time is on the x-axis. And what you see is for the first 100 milliseconds, there is no difference between figure and ground, so the modulation is 0. But at 100 milliseconds, we start to see increased spiking in the deep layers and superficial layers, and not so much in layer 4. So we see this U-shaped profile with higher spiking activity in superficial layers and deep layers. Now, that's true in the spiking. It's also true in the current source densities. In this case, the two techniques actually agree with each other. So if you look at the current source density difference between the figure and the background, the difference in current flow, then again, you see nothing for the first 100 milliseconds. Then at 100, when the spiking increases, we see a sync source pair in the superficial layers, in layer 1, and a sync source pair in the deep layers as well. And so these sinks in layer 5 and layer 1 are exactly the layers we would predict them to be from the feedback projections to V1. Okay, so we see this in general, a V-shaped profile for top-down process with weaker activity in layer 4, with weaker modulation in layer 4, and stronger modulation in the superficial and deep layers. And how does that compare to fMRI studies? Well, there has been a Laminar fMRI study looking at the exact same paradigm that we just looked at. But there have been 2 studies which have looked at these top-down processes, which I'm going to examine a little bit in more detail. So this study from Koch et al. From Flores de Lange's group looked at Knizia figures. So here we infer the presence of a triangle from the alignment of the inducers. And here the inducer has been rotated to break the illusion. And they looked at the bold signal in V1 from this region here, where there's actually no visual stimulation. So there's just a gray screen, but this region is either part of this illusory triangle or it's part of the background. And if you look at the laminar profile of these 2 conditions, then what you can see is that you see a general slightly V-shaped profile actually, but the biggest difference between these 2 conditions is in the deep layers. Right? So you see a strong modulation of deep layer bold signals when there is a Knizia figure present. Okay, so that's a little bit different to what we just saw in the EFIS studies, where we saw more of a V-shaped profile with also modulation in the superficial layers. And I'll come back to why that might be in a couple of slides. Here's another study also looking at top-down influence on visual activity. So here, this is a study from Muckli et al. They showed 3 different natural images. And in some of the cases, they occluded the bottom corner of the image with a white square. And then they looked to see whether they could actually decode the actual identity of the image from the information from voxels which were responding to this part of the space. Now, as you would expect, if you use unoccluded parts of the image to do the decoding, you get very, very good performance. That's the red line here in this laminar plot. These are deep layers, these are superficial layers. And the decoder could decode which image was present in the sort of high 70s, 80%. However, if you use occluded regions of the scene to build your decoder, then you actually see your chance in the deep layers, and it moves, it gets strongly better as you move towards the superficial layers. So in the very most superficial layers, you actually get good decoding. Okay, so here this shows kind of the opposite profile to the previous study. Again, it's a top-down effect. The V1 is receiving information here about what is present in the rest of the scene, which you can decode, but it's really only coming, you can only really decode it well from the superficial. So neither of these two studies show this kind of V-shaped profile that we see in electrophysiology. Now, there could be, of course, because in both of these studies, they didn't actually have visual stimulation in the part they're recording from, whereas in our study, we did have visual stimulation. So we recently had a look at a sort of paradigm where we also removed the visual stimulation, but still had a top-down effect. So this, we're moving to mice here, but the principle is pretty much the same. We had a laminar electrode in V1. We have head-fixed mice who are looking at visual stimuli on the screen. And in particular, I'm going to be focusing on the difference between conditions where we have visual stimulation, such as this figure or uniform condition, to a condition in which we had a gray circle presented on the screen. So the receptor field would be in the middle of this circle, and we would call this either the occluded or the gray circle condition. Now, we don't know what the mice perceive here. They might see this as a gray circle on a background. They might see it as a hole. They might see it as an occluder. We don't know. But what we do know is there's no visual stimulation present in this particular point of visual scene. So you could ask, do we actually see a response to this gray circle? Okay, so there's nothing happens in the receptive field, but we do see a response. So here we see a receptive field map from an example cell in V1 of a mouse. The black circle here is the location of the receptive field, and the white circle is the size of one of these figures or gray circles. So you can see we have centered this circle nicely on the receptive field. The receptive field is entirely within the circle. Here's a typical profile. This is multi-unit activity again. This is time. You see when there is a visual stimulus, and it doesn't really matter if it's a figure or a uniform stimulus, you get a strong visual response. What's interesting is the red condition. So in the red condition, you can see the response is actually delayed by about 100 milliseconds, and then it starts to increase. So we do see spiking, but it's delayed relative to the visual conditions. That was true in this example. It was true across the population. So here you see the population activity. You can see, again, this delayed activity in the gray circle condition. But towards the end of the response profile, it actually reaches a level pretty similar to the uniform condition. Okay, so it produces strong spiking. Let's have a look across the layers. So now I'm just going to compare the uniform condition to the gray circle condition. And in the uniform condition, we see a similar laminar profile to what we saw in the monkeys. So a strong peak of activity in response to this grating. This is layer 4 in the mice, so it's a lot smaller than it is in the monkeys, so it's just a couple of contacts. But in the gray circle condition, we really see a very dramatic difference. So there's this much weaker activity in layer 4. Right, you see very strong response in the deep layers, a reasonably strong response in the superficial layers, and almost nothing in layer 4. And there's been a recent, I think last week, there was a Nature paper from the Scanziani group showing that this gray circle response, this response to this occluder, is coming from feedback from higher visual areas. So this is not a feed-forward response, this is a top-down. Okay, so this just shows you the laminar profile of the effect. It's much stronger in the deep layers than it is in layer 4, superficial layers being somewhere in between. So here we're starting to get a little bit more towards what was observed in the Koch study with the Knizia triangles. So there they also saw a sort of V-shaped profile, but it was biased towards the deep layers. And we're also seeing a V-shaped profile, which is biased towards the deep layers as well. So this suggests some promising agreement between the EFIS and the bold studies of top-down processes. Okay, so coming to the final part of my talk, I just want to look at one more top-down effect, which is the effect of attentional selection and working memory. And this is all work carried out together with Timo van Kerkhove. So we used a paradigm which we use a lot in the lab to measure attentional labeling. That's a paradigm called curve tracing task. Here we have the monkey fixate. We have a receptive field mapped out in V1. Then we show these stimuli, these curves. One of the curves is attached to the fixation point, and one isn't, and there are some distractors. The monkey has to keep his eyes on the red dot, but then mentally trace that curve. And then when the dot disappears, he's meant to make a saccade to the target at the end of the curve. Okay, so you can set things up so your receptive field is on the target curve, the one that's being traced, and we know that monkeys actually spread their attention along the curve. Okay, so this would be an attention in condition, or we could put the receptive field on a distractor curve. So here the monkey's actually tracing this curve, and now this is the attention out condition. Now, we get a typical sort of response profile. You're probably getting used to these now, where you see a later response to the target above the distractors. You see a late modulation spiking. We compare this to a memory case where the monkey's doing exactly the same task, so he has to trace this curve, but the curve is only presented briefly. It disappears after 150 milliseconds. The monkey then has to maintain the target curve in memory. We present a mask to get rid of any sort of ongoing neural activity, wipe it out. And then there's another delay period. And finally, he's given a cue, which tells him to make a to one of these two targets. So he has to remember which one was connected and make an eye movement to the target. Again, we're interested in the activity while the monkey is maintaining the target curve. What you see is, so we have the visual response to the onset of the curve here, and then offset when the curves disappear. Then you get a period where there's nothing on the screen, but there's still a higher response when the monkey is tracing a target curve compared to the distractor curve. Okay, so when the receptive field is on the target curve, you get more activity than when it's on the distractor. Then the mask wipes out the activity completely, so all differences between conditions are gone in this period. And afterwards, again, there's nothing on the screen. We're in delay period 2, but there's still a sustained increase in activity when the receptive field is on the target curve, even though the target curve is not visible. Okay, so we seem to have this working memory trace present in V1 during this delay period. Now we can look at the laminar distribution of that modulation, and what we see is both for the attention and the memory case, we have a U-shaped profile of spiking modulation. So this is attention when the curves are actually on the screen, and you see there's more modulation, and it has this dip in layer 4. Where for the memory case, if we just look at this last delay period 2, where the memory seems to be reinstated in V1 after the mask, and we see this very clear U-shaped profile. The modulation is almost only in the superficial and deep layers. So that suggests that these cases where we don't have a visual stimulus on the screen, they'll be very sensitive measures, very sensitive to laminar differences, more than when there's actually a visual stimulus present. Quickly comparing that to an fMRI study, which used a similar kind of paradigm. This was a working memory study, also from the DeLange group from St. Lawrence. Subjects had to remember an orientation of a grating. They presented 2 gratings, then a cue. The cue here, number 1, means they have to remember the first orientation and forget the second one. Then there was a long working memory period followed by a probe. And here they just had to say, is the probe the same orientation as the one they're remembering? And this was staircased to make it challenging. Then I'm going to ignore the time course for now. I'm just going to look at the difference in neural activity when, so what they did is they actually split the voxels. They found voxels which strongly preferred one of the 2 orientations, either the clockwise or the counterclockwise orientation. And so they were able to look at a voxel's response when the preferred stimulus was being held in memory compared to when its non-preferred stimulus was being held. Right, and you actually see that there's an increase in activity when the preferred stimulus is being held in memory. And that difference between preferred and non-preferred showed a V-shaped profile. Right, so this is a laminar plot, deep, middle, and superficial layers. You see that the difference between preferred and non-preferred orientations in memory is strongest in the deep and superficial layers. So this really strongly resembles the laminar profile that we observed in our working memory task in monkeys. There's really good agreement here between the laminar fMRI and the spiking. Okay, so just to sum up, we've looked at 2 very different functions of visual cortex, where the layers play quite different roles. Visually driven bottom-up activity, we drive visual cortex with stimulus, which creates a very strong activity in layer 4C with activity descending or sloping away from layer 4C. And that was true both in the spiking activity and in the laminar bold signal. If you look at the feedback or the top-down processes, so if you look at figure-ground modulation, if you look at attention or working memory, then you typically see a U-shaped profile in the spiking with stronger activity in the superficial and deep layers. And that is partially observed back in fMRI studies. So some fMRI studies find more activity in deep layers, some more in the superficial layers. And then the last study we looked at, they actually covered the full V-shaped profile of this response. So what we're seeing is a very promising start for laminar fMRI. It seems to be particularly sensitive to these feedback or top-down processes, and it seems to agree quite well with the spiking activity observed in EFIS. It may be therefore a really useful tool to actually look at these cognitive processes which modulate V1 activity. And finally then, just the acknowledgements. I'd like to thank Cor and Annika, who are the animal technicians who worked with us throughout this study. Timo van Kerkula, who recorded the working memory study we just looked at. Lisa, who recorded all the mouse data. And all this work was carried out in the lab of Peter Wolfsomer. And thank you very much for listening. Thanks.